In this video, we'll be reviewing central dogma, which is the expression of DNA to protein. This includes DNA replication, so DNA making more DNA, the exact copy, transcription, which is DNA to RNA, and translation, which is RNA to protein. Starting with DNA replication, some reminders about the enzymes involved. So there's helicase, which opens the double-stranded DNA. There's topoisomerase, which helps with that opening by kind of like wiggling the DNA. There's DNA polymerase, which actually does the job of matching the bases of the growing new DNA strand with the template and original strand. So for example, if this was an A, DNA polymerase is making sure that a T goes across from it. Bases have to be paired up with a hydrogen bond. And there's also primase, which puts an RNA primer down so that DNA polymerase can do its job. DNA polymerase can't start on a naked piece of DNA. It needs an RNA primer to get going. There's also ligase, which fills in the breaks between Okazaki fragments. Remember, there's a leading and a lagging strand. The leading strand is the strand that's being replicated towards the replication fork. And the leading strand is always going to be with the 3' side of the new strand facing in, because DNA is always added to the 3' side of a growing strand. The lagging strand is the strand where the 3' is facing away from the replication fork. And in the lagging strand, as helicase opens the DNA up more, primase and DNA polymerase have to go back and fill in that newly exposed spot. So in the lagging strand, that's where the Okazaki fragments are. So as a reminder, A pairs with T, C pairs with G, and this is just one side of a replication fork. When DNA is replicated, it's replicated from an origin of replication in both directions. So the leading and lagging strands really depend on which way the DNA is opening in the section that you're looking at. Next up, transcription. So in transcription, we're going from DNA to RNA. So RNA polymerase does pretty much all the work. It opens the DNA and it matches the base pairs of RNA with the DNA in the template strand. So here I have for you a pre-mRNA that's being made. And you can see here A is paired with U, T is paired with A. Remember, in RNA, there's no T. It's U in the place of T. And you can see that we're, again, always adding to the 3' side of the RNA. So the RNA polymerase is moving in this direction, and it's going to add on a new RNA molecule. So what would it be adding next here? It would be adding a G, and then another G. So it's growing in that direction. So you'll notice that the template strand is the opposite of the pre-mRNA, right? Because we're base pairing here. So we call this strand the coding strand because it has the A, that should be a T, A, T, G, C, C, which is what we're seeing on our pre-mRNA, A, U, G, C, C. So the coding strand is what we're trying to make, and we have to use the template strand to make it. After the pre-mRNA is made, RNA processing needs to happen, which includes the removal of introns, and we keep the exons. Also, a poly A tail is added in a 5' methyl cap. These modifications help the RNA be able to leave the nucleus and also help it last longer when it gets into the cytoplasm. A reminder, alternative splicing is possible because these exons can be arranged in different orders. So say this is exon 1, 2, 3, and 4. They could be arranged in that order, or it could be 1, 3, 4, 2, or 1, 2, 4, 3. And depending on that order, that's going to result in a different amino acid sequence and so a different protein. So that's how we have fewer genes than the number of proteins that we're able to make. Next up, translation. This is when we're going from RNA to protein. So reminder, ribosome is the molecule that is doing this, that is taking us from mRNA to protein, and tRNAs come in and bind with codons, which is a set of 3, and the tRNAs have an amino acid attached to them based on that anticodon. So this is how we get the correct amino acid paired up based on the sequence of the mRNA. The ribosome would be moving this way. I have my start codon here, AUG, that coded for methionine, and that MAT you can see is already added. So when this is fully translated, we'll have a long protein chain with amino acids in a certain sequence. And remember that protein is going to fold in a certain way, and that's part of what gives it its function. An additional reminder, in prokaryotes, transcription and translation can happen at the same time because the DNA is not in a nucleus. So this RNA polymerase can be copying the DNA into RNA at the same time that a ribosome is bound to that RNA, making a protein. But in eukaryotes, the RNA needs to be fully made first and leave the nucleus before it can find a ribosome to make protein.